everyone. In this video, we discuss about desensitization of beta-adrenergic receptor. What is desensitization? We have already discussed about desensitization in the case of acetylcholine receptor. When a signal, here it is epinephrine, persists for some time, what happens? The receptor is desensitized. How? Here, this process is mediated by two proteins, beta-adrenergic protein kinase and beta-arrestin. Here you can see the beta-adrenergic protein kinase is represented in yellow in color. This one is beta-adrenergic protein kinase. And that this red one is beta-arrestin. This beta-adrenergic protein kinase is a member of G-protein coupled receptor kinase. That is, this beta-ARK is a member of GRK. These are the two proteins involved in desensitization of beta-adrenergic receptor. In epinephrine signaling, what happens? Epinephrine bind to receptor, receptor bind to G protein, and the alpha subunit of G protein dissociated from gamma and beta, and alpha subunit bind to enzyme, and enzyme produces cyclic AMP. Then glycogen is breaking down into glucose. After that, what happens? The epinephrine is already bound to this beta-adrenergic receptor. Here you can see epinephrine is bound to beta-adrenergic receptor, and here no alpha subunit. Where it went? Alpha subunit binds to enzyme, and enzyme produces cyclic AMP. And this gamma and beta subunit of G protein recruits beta-ARK into the plasma membrane. And this beta-ARK phosphorylates serine residue where at the carboxyl terminus of this beta-adrenergic receptor. And this phosphorylated site create a binding site for beta-arrestin. Two steps: beta-ARK phosphorylates serine residues at the carboxyl terminus of the receptor, and this provide a binding site for beta-arrestin. And here you can see the beta-arrestin binds to phosphorylated serine residues of beta-adrenergic receptor here. And what is the function of beta-arrestin? This beta-arrestin prevent interaction between receptor and G protein. So there won't be any interaction between this beta-adrenergic receptor and G protein. Okay, and one more function. The receptor from plasma membrane is removed by endocytosis and to form a small intracellular vesicle. You can see a small intracellular vesicle. It is known as endocytic vesicle. The receptor in the plasma membrane is removed by endocytosis to form endocytic vesicle. And in the vesicle, this arrestin dissociates. You can see arrestin dissociates and then it recycled. And this receptor is dephosphorylated and returned to cell surface for next round of action. This is all about desensitization of beta-adrenergic receptor. If you like this video, please share and subscribe. Thank you.